Good afternoon. I'm Richard Kincaid, and I'm the CFO of Healios. At Healios, our mission is to transform lives through the power of regenerative medicine. From our base here in Tokyo, Japan, we're leading the world in the development of cell therapies to treat intractable diseases. We've been at this for a while. Our founder and CEO, Dr. Hardy Kagimoto, set up the business in 2011. In 2015, he listed it on the Tokyo Stock Exchange, and today we have over 150 people based in our Tokyo and Kobe operations. And over these 10 years, we've built up certain key strengths that position us well to be a successful regenerative medicine platform company. One of these is our strategy, where we combine a near-term commercial revenue opportunity with a licensed-in product here in Japan, with the development of a world-leading iPSC technology platform. Importantly, we're doing our initial work in Japan, which has the most favorable regulatory framework in the world for regenerative medicine. So with this as a backdrop, we've recently completed 2 clinical studies using multistem for stroke and ARDS here in Japan. We've read out positive top-line results for ARDS, and we're now pending stroke results, which we'll get to through after a requisite follow-up period over the next several months. Now, while we've been making all this progress with our multistem development in Japan, we've also been advancing our iPSC technology platform. One of the key recent successes was last year we established a clinical-grade universal donor iPSC line, which we call our UDC line. That's an important starting material for various innovative products we're building here at Healios with our iPSC technology platform. And so we have pipeline assets across a range of domains, includes immuno-oncology, ophthalmology, and organ buds. And supporting all this is a deep and diverse team, manufacturing capabilities that are both in-house and through partnerships, and strong financial resources. A bit more about our strategy. I mentioned we're developing multistem in the near term here in Japan. It's a bone marrow-derived somatic stem cell product. It's a MAPC, a multipotent adult progenitor cell product. And we're in the most advanced stage of developing this for both ischemic stroke and ARDS. And so we're using what comes from this in terms of it being a commercial revenue opportunity and ultimately will generate cash, valuation uplift. And also, with this, we're building out a commercial apparatus. We're becoming a commercial pharmaceutical company here in Japan. That gives us muscle to do many things. So we're leveraging all of that to build out this iPSC technology platform. There are multiple pillars to this today. And just as background, we've been in the iPSC therapeutics development business for a long time. We were the first in the world to put an iPSC-derived therapeutic into humans. This is back in 2013. So we have a long history with these cells and capabilities with iPSCs. And early on, we learned the importance of moving towards a universal donor cell construct. And so that is something we started about 6 years ago. And I mentioned the universal donor cell line that we created last year. And so we combine iPS cell capabilities with gene editing expertise to make engineered iPSC products. And that then extends into a next pillar, which is becoming a platform within the platform. These are engineered iPSCs that we're using to make immune cells to treat cancer. And our lead iPSC product today is a functionally enhanced NK cell that we're using to treat solid tumors. We've also got strong manufacturing capabilities, CMC capabilities in-house, and then through partnerships, very close corporate partnerships that we have. And so taken together, this puts Healios into position to make pipeline assets that we can take out to the global market across a range of domains, as I mentioned. So to recap, we have very strong product creation capabilities here in-house at Healios. We have equally strong CMC manufacturing capabilities. And this is now a set of resources and capabilities that we're going to use to take products to the global market. Increasingly, you'll see us talk about our development efforts towards the United States. Now, I guess as a footnote, we have a venture fund arm to the business as well. And maybe this demonstrates a little bit about our reach, but we set up a venture fund in January of this year with our capital and some external capital. And this fund invests in bioventures in both the United States and in Japan. It's a return-seeking fund, but for Healios, it's important because it makes us smarter, and it leads to connections with innovation and innovators around the world. This is ultimately going to create business opportunities for Healios over time. So now briefly on the regulatory framework here in Japan. There's a system in Japan called the Conditional and Time-Limited Authorization System. And what it allows for with a regenerative medicine product, you have to prove safety like you do everywhere else. But as long as you demonstrate a trend of effectiveness, you may be able to apply for early approval and for up to 7 years sell your product and get reimbursed while you continue to build out your data set. And this is de-risking of the development process for regenerative medicine products here in Japan. It allows Healios to take these on with less cost and less risk than would be the case, say, in the United States. So our pipeline is made up of 3 broad groups. We have inflammatory conditions, immuno-oncology, and replacement therapies. In the inflammation space, we're treating acute inflammation in relation to ischemic stroke and ARDS using multistem. As I mentioned, we're in the most advanced phase of developing this product here in Japan for these indications. We've completed enrollment for both studies. We've released data, top-line results for ARDS. And with respect to stroke, we're pending data. Now, we have Sakigake designation for stroke here in Japan, and that allows us to do a rolling submission. We've already submitted the non-clinical regulatory package. We've submitted our CMC package, and now we need to move for both a stroke and ultimately for ARDS towards building out clinical packages to provide to the regulator. So that's where we stand with stroke and ARDS, and this is very near to market. The next important area for us is immuno-oncology, where, as I mentioned before, we're building out a functionally enhanced NK cell for solid tumors. So this product code is HLCN061, and we're increasingly putting a lot of effort and focus and resources into this, and we're going to be putting more information out into the market here in the coming months so that stakeholders can gauge this product and where we stand versus the other peer products that are out in the market. But our target is to build a best-in-class NK cell to treat solid tumors. On the replacement therapy side, we've been working on this area for years. We have great resources and abilities to enable us to make successful replacement therapies. There are a couple in the pipeline today, but we have a lot of research assets in this space too, and we're advancing the ball in several areas. So just briefly on multistem. So, if there are waves in the regenerative medicine space, and when you think about timing, was a product ready to bring to the market? We think this is one area and the type of product, especially here in Japan, with the regulatory environment. It's really ripe and ready to bring forward into a commercial state, in our market. And there are a lot of reasons for that, right? There's proven record of safety. There's indication of efficacy in trials that Atherosis has run. We now have that from our ARDS study. This product is easy to store with a long shelf life. It can be stably frozen. It's an off-the-shelf product, and it's very easy to administer. We thaw it, put it in an IV, and administer it over the course of an hour in the acute care setting. So a lot of reasons why this is pretty straightforward for us to commercialize, especially in a relatively contained market in Japan. And so we ran a study for ARDS, which has 2 cohorts. Cohort 1 was the original cohort. This is prior to COVID-19 coming about. We had 30 patients in an open label study to treat 20 with the product, 10 with standard therapy. Our primary endpoint was ventilator-free days at 28 days. We had some secondary endpoints around mortality at various points in time. With the cohort 2, which is just 5 patients that we treated who have COVID-19 induced ARDS, it's a small group with a primary endpoint just of safety with some secondary endpoints similar to what we're doing for cohort 1. And this is the data that we achieved in that study. It's just top-line results. And you can see here for cohort 1, we had a 9-day pickup in ventilator-free days for the treated group. Standard therapy group, the number of days out of 28 when these patients were off a ventilator was 11. We had 20 out of 28 days where the patients were off a ventilator in the treated group. These are median numbers. So that's very encouraging, as is the mortality data, where we had a 39% reduction in mortality as compared to the standard therapy group. In cohort 2, there were no safety issues. This was just 5 patients again. The ventilator-free days median was 25 days. All the patients were off a ventilator within 28. None of them died. 3 of them were off within 3 days. So while it's a small group of patients, it was encouraging data. That's the data we got in our ARD study. This is data from Atherosis' phase 1/2 double-blind study in the US with patients. This is all cause ARDS. Our study was in patients with pneumonia-induced ARDS for cohort 1. But it's similar data to what we got. And so our data is validating of the Atherosis data. Taken together, they demonstrate a clear trend of efficacy. And, you know, with ARDS, it's in Japan about a 10,000 patient indication, at least as is currently assessed. It's an orphan indication. We received orphan regenerative medicine status for this in Japan. A lethal indication. There are no other treatments for it. The data that I just showed you, we'll be combining the two as we bring it forward through the regulatory process here. We are looking towards seeking out an approval, making a filing as soon as we can to get this product brought to patients as soon as possible here in the Japanese market. So now moving on to stroke. This is just a slide showing our timeline. We enrolled all the patients as of very recently. This is a big study. It's 220 patients, double-blind study. The primary endpoint is excellent outcome at day 90. It's a very difficult hurdle to meet. And then we have certain other data points that we will work towards at the 1-year mark as well. And so with this study, as I mentioned before, we have Sakigake designation. And we've already submitted 2/3 of the regulatory documents that we would need to, 2 out of 3 of the packages, but non-clinical and CMC package. And now we're working towards ultimately having clinical data and would be submitting a clinical package if we get the data that we want. So that's going to play out over the next several months. So while we don't have our data yet, this is Atherosis data from their phase 2 double-blind study in the US. This is post-hoc data in patients that were treated within 36 hours of stroke onset at both day 90 and at the 1-year mark, for severe ischemic stroke patients using an excellent outcome primary endpoints. Same parameters as our study. And you can see both at day 90 and at day 365, they had statistically significant outcomes. Again, this is subset analysis for patients that were treated in that time window within 36 hours. And so as we work towards ultimately having our data, what are we gunning for? We would like to get a significant difference in the number of excellent outcomes in that treated group versus placebo group, and hope to show statistically significant results. So we'll see that when we are able to open up the data and find out if that's what we've got. So now if we do get good results, it's very important because stroke is a big indication. About 300,000 patients per annum in Japan. Our focus is on severe stroke patients and those that can be treated within 36 hours of stroke onset. And that's a big market today that's not addressed with anything. TPA is a clot dissolving agent that exists within about a 4-hour time window here in Japan. And you can get mechanical reperfusion, physical removal of the clot within about an 8-hour time window. And if our therapy is successful, we'd be able to extend that time window out to 36 hours, treat far more patients than have the ability to be treated today, and improve outcomes, bring something that's maybe more effective than what is in existence. Because a lot of these TPA and MR patients end up failing in the treatment, and then post that they don't have an available therapy. So that's what our target is with this, to bring impact to these tens of thousands of patients in Japan with this regenerative medicine product. Now, I didn't mention pricing, with respect to stroke and ARDS, but I'll just say this. So 60,000 patients or so, it's the target market for stroke. It's 10,000 for ARDS, 70,000 patients in Japan. Products like this that have emerged. There's one for GVHD, for example. The pricing has been in the $100,000 to $150,000 range, and so it doesn't take a lot of penetration into this market in Japan for it to be a very important source of revenue ultimately for Healios. So with that. All that being said, that's the status of the multi-stem programs today. Our domestic development, our build of Healios into a commercial biopharma in Japan initially. The first step is around this licensed in product here in Japan. And I'll segue now into our iPS cell platform and programs to give you an update on that. So I mentioned our universal donor cells, and I just want to briefly explain the why behind it and where we stand. So as I mentioned before, we learned early on that we needed to have a universal donor cell line to really industrialize iPS cell therapeutics. Right. So for years, we've been working on a universal donor cell line. We established the clinical grade line last year. The goal is to create a starting material that can be used to make iPS-derived products that are off the shelf, that are cost efficient to make, that are it's one product for all patients, and that don't require immunosuppression because they don't invoke an immune response. That's the goal, right? And so that's what we are setting out to do. We have our initial clinical grade line. There are other versions that we may roll out over the course of time. But this particular line we've taken to the FDA and the PMDA. There are no concerns in relation to use of this through phase 3 trials. And this readily differentiates into all these cells that we make here at Healios. I'll go through a couple examples in these slides. And we have active engagements with several companies and academic institutions in relation to using this as a starting material to make therapeutic cells that they're making as part of their cell therapy activities. And so how do we do it? We're knocking out class 1 and class 2 HLA molecules. We knock in certain genes to improve immune evasion. And we also knock in a suicide gene as a safety switch. And these are what the cells actually look like. This is a picture of the universal donor cells. And those can then be further differentiated into various cells that retain these universal features to create products that are broadly applicable across all patients. And I'll very quickly go through this. This data is just sort of initial data that we got, maybe a bit data we have more now, but just shows expression of genes and proteins as a result of the changes we're making, where we're knocking out certain HLA expression. We get the knocked out status here through these pictures. And here we're adding certain genes to improve immune evasion. You don't see expression of those genes here. Well, with the knock-ins, you can see expression. So that's just demonstrating the basic concept of what we've done. This is not all the data, but is exemplary. Now, with respect to karyotypic aberrations, there are none. These are iPS cells. They maintain pluripotency. As I said, we can readily differentiate them into all the cells that we make here at Healios. These are just a couple examples. With the suicide gene, we've tested it in mice, we've tested it in vitro. We're able to, with a simple medicine, cause these cells to go apoptotic, and so that's been tested thoroughly as well. So that's just an introduction to the UDC. It's part of the iPS platform. There are many things that go into the iPS platform. It's an important starting material. And so I'll give a couple examples in a little while of UDC-derived products that we're working on. But before I do that, I want to go through our NK program. We're focused in the near term on building out a multi-stem franchise here in Japan, but we are increasingly putting our resources into the development of NK cells for solid tumors. And the reason why is it's the leading cause of death in Japan. Cancer is the #1 cause of death here. 90% of those deaths are due to solid cancers. CAR T has been successful at treating blood cancers, has not been the case for solid cancers. These patients need a better treatment, and they need it as soon as possible, and we're working towards that. And so I mentioned we're developing a best-in-class, functionally enhanced NK cell for solid tumors. We edit into the iPS cells the functional enhancements that we're seeking to have at the NK cell level. We then differentiate these into NK cells. We've made a master cell bank for this product, and can efficiently manufacture NK cells now in 3D bioreactors. And so we're very advanced now with this. And these NK cells then are very powerful cancer killers. And I'll go through some slides to start to demonstrate that with some early information that we're releasing here. So rather than talking through it, I'll show you through a video, a few videos here, what these iPS-derived NK cells do. And so you can see there's a little bright spot here. This is an NK cell sitting on top of a cancer cell. These are this is a lung cancer cell line. So if I show the video, you can see the NK cell attacking away at the lung cancer cell, and it gobbles it up and then moves on to the next one. And this is just a simple illustration of what these NK cells do at the sort of most micro level, sort of NK cell, one NK cell versus one cancer cell. But then this is a cancer spheroid, and we're showing here migration. So there are a lot of dots up here. These are NK cells. There's translucent specks down here. This is a group of NK cells too, and you can see that they're going to be moving to the right, and then ultimately, when they recognize the cancer, they, as a group, move towards it and attack it. So I'll just play the video. You can see them moving to the right, and then all at once swarming and going towards the lung cancer spheroid and attacking it and trying to kill it. So moving on, here we're showing invasion. All these pink specks, these are NK cells. These are, again, all NK cells we make here at Healios from iPS cells. And this is another, again, lung cancer spheroid, and we'll see NK cells, see many of them go inside. And they attack these cells from the outside, they attack the spheroid from within, and that is part of how they do their work at destroying the cancer. And now, just to finish these videos off, you can see Healios-derived NK cells attacking this cancer spheroid, again, from the outside and ultimately from within. And this, in this case, the cancer gets overwhelmed, it gets destroyed by the NK cells. So you can see them destroying the cancer, and ultimately all the cancer cells go apoptotic as it explodes. So, we haven't released much on the profile of the cell candidate yet. We're getting real close to being able to do that. And we're getting close to being able to put animal data out there as well. So that's forthcoming. But the point I'll make here is that we've made several functional enhancements to these NK cells. We think we have the most comprehensively enhanced NK cell available. We also think our starting point is very potent. Just looking at unedited iPS NK cells that we make here are very strong relative to data we see out there in the literature. And so we're excited about this product. We're excited about it being differentiated versus some of the peer products that are out there. We look forward to putting more information on about the actual edits that we've made and getting some animal data out there soon, and then ultimately development plans. And to that point, we're working on PDX mice with the National Cancer Center of Japan. These are human, mice with human solid cancers. That's some of the testing we're doing. We're doing CDX mice testing in-house, and we have some other relationships with KOLs with models here in Japan, and we're moving the testing forward in a multi-pronged fashion. And as I mentioned before, we can already make these efficiently in 3D bioreactors. We developed that technology in our process development lab. We are now setting it up in a CPC that is in Kobe. And this particular facility is going to be able to supply these NK cells for any clinical trial we would run, whether it's here or whether it's in the United States. It also can be, it's a process that can be scaled out or scaled up to deal with commercial manufacturing. So we're quite pleased with how far we've advanced the manufacturing capability for these NK cells already at this relatively early stage. So that's where we are with the NK cells. Now, this is a UDC derived product that I'll mention, a separate product on the IPS side of our business. And it's probably as far as we know, the first universal donor iPS cell derived photoreceptor replacement therapy in the world. So we've created what's a pure cone photoreceptor product that we think has certain advantages to it. We haven't released a lot on this yet. To be able to replace this layer of cells in eyes that are damaged by certain diseases of the eye. So for years, we've been working on RPE cells, and we continue to move that forward. Now we have this UDC-derived photoreceptor, and soon we'll be be in a position to release large animal study data for this as we sort of build out this early POC that we have for this particular cell type. It's one example of something we make from a universal donor iPS cell. And there are other examples. For example, we released some information recently that we have derived from UDC pancreatic beta cells as well. And while we don't have this on a UDC platform yet, we are moving towards, through our academic partners here in Japan, first-in-human proof of concept testing for liver buds for metabolic liver disease. This can be put into children with OTC. And this is a technology that we've been working on for a number of years. We've made some very big advancements on the manufacturing side recently. I'm just going to show you a video just for what it to show you what it is. Where we combine liver precursor cell with vascular endothelial cell and MSC to make a functional mini liver that autonomously vascularizes and integrates with surrounding tissue and implanted into an animal. And so you can see here the 3 cells being mixed in a certain way, coming together and forming a mini liver like this. We can then put these into mice or humans. And this is the results of a mouse experiment, which is some time ago now, but we have animal POC in this, or the mice that got the liver buds survived, the mice with liver disease. Those that did mostly died off very quickly. Ultimately, putting this onto a universal donor cell platform is going to make it a potential treatment for things like cirrhosis. And so we're moving the ball forward on this and other programs as well to put them onto a universal donor cell platform. So with all that being said, so we have various programs here. The focus near term is on multi-stem. The other focus is on bringing these NK cells forward. We're pushing really hard to move them forward quickly. Our team is diverse in their skill set and background, and it matches the strategy that I've talked about today. We have team members, most of whom come from Japanese pharma, and they put us into the position to build out this commercial biopharma company that is commercializing a product for meaningful indications like ischemic stroke and ARDS in Japan. And then we have other people who are very entrepreneurial, have cell therapy backgrounds, who in many cases are foreigners, that put us in a position to build this bridge, where we're taking product creation capabilities of Healios and the CMC manufacturing capabilities of Healios, and we're going to deliver those products ultimately to the United States. And so we're positioning the team increasingly to be able to do that as well. And on our capabilities, we have over 90 people in our Kobe Research Institute, over 30 PhDs. This is where a lot of the innovation at Healios is coming from. And we have several facilities, research facilities in Kobe, in a particular neighborhood there. And as I mentioned before, we now have a CPC. Importantly, on the manufacturing side, one of our important manufacturing partnerships is with Nikon. In addition to them making multi-stem for commercial purposes here in Japan, they're also a capital partner of ours. They're one of our largest investors. So we've worked closely with them, and our partner Atherosis has worked closely with them to get multi-stem production ready for commercial over the last few years. Now we're in the final stages of preparing that product for commercial manufacturing here in Japan. And we've always had strong financial resources at Healios. Hardy founded the company 10 years ago, did one A round, and then listed the business in 2015. We've raised over $300 million since inception. Our market cap is over $1 billion. We have around $100 million of cash as of the last disclosure date. We spend about $10 to $15 million per quarter, but it's really been closer to 10. We spent 40 last year, spent 40 the year before. And so relative to cash burn, we're in pretty good shape when it comes to cash position. And we have a lot of important events coming up over the coming months. So in conclusion, I put the strengths back up here, and I won't go through it line by line, but just to repeat, our strategy is important because we have near-term shots on goal to become a commercial biotech company here in Japan. And we're using what comes from that to be stronger and more efficiently build out a world-leading iPSL technology platform. We're starting here in Japan. We think it's an advantage to do so because the regulatory framework for regenerative medicine in Japan is very favorable. We're not limiting ourselves to Japan, but we're using our strength in our home market to ultimately build ourselves towards clinical development capabilities in the US with our products that we're making here in our platform. And we have various advantages on that side of our business as well, where we're in possession of an innovative universal donor iPSL line, technologies that make us capable of building on that. And within our iPSL technology platform, we have very potent NK cells that we're moving forward for solid cancer indications, and ultimately we're going to develop that on a global basis. And so we're doing a lot here at Healios, but we have a lot of resources to support those efforts. We've got a deep and diverse team, really strong manufacturing capabilities and partnerships, and we've always been in a strong position financially. So that's all I'd like to say today. Thank you so much for listening to the Healios story. We appreciate your support. With your ongoing support, we're going to be able to bring innovative regenerative medicine products to patients to cure disease, and we look forward to doing that as fast as we possibly can. Thank you so much for your time.